So I'm going to give a brief introduction because I really need to get out of the way because I know you're not here to listen to me for sure, for our next speaker. Probably an astronaut who really doesn't need an introduction, but I'm going to give you a little bit of information about her background. Dr. Peggy Whitson, who most recently flew on Expedition 5051, and I think there were more than that actually, she'll correct me, and participated in 4 spacewalks, bringing her career total to 10, with a total of 665 days in space. Dr. Whitson holds the U.S. record, placing 8th on the all-time space endurance list. She's an Iowa native, also completed 2 6-month tours of duty aboard the stations for Expedition 5 in 2002 and the station commander for Expedition 16 in 2008, where she accumulated 377 days in space between the 2 missions, and I believe was the first female commander at that time. So clearly a record breaker in many ways, and an impressive individual in many ways. Just the contribution she's made, not only to space flight, but Dr. Whitson has been a principal investigator. I mean, she is a scientist among scientists, you know, and one of the giants, the shoulders that we will stand on for a long time to come. As I said, she's a not Iowa native from Mount Ayr, Iowa, hometown Beaconsfield. She is married to another principal investigator, Dr. Clarence Sams, who is pretty indispensable. We pull on Clarence all the time in many ways. Says she enjoys weightlifting, biking, basketball, and water skiing, among her many other talents. She graduated from Mount Ayr Community High School in 1978, received a Bachelor of Science in Biology and Chemistry from Iowa Wesleyan College in 1981, and a doctorate in biochemistry from Rice University in 1985. And really, I just want to welcome Dr. Peggy Whitson to this stage and let her help us understand what it's like to be a world record holding astronaut in spaceflight. Let's welcome Dr. Whitson.
Thank you. Very nice. Well, I am really happy to be here and talk to you guys. I had a fantastic time on this last mission, and I wanted to share some of the parts of it that were really exciting to me. And we'll have some time at the end where I can take questions and hear about anything that you might be interested in. The biggest thing about doing research in space is it's in space, and there's 0 gravity. That's one of the key principles of why we do research in 0 gravity, but it also has its complications as well. Whether that's the hardware or losing the tools, it can add complications to what we do in space. I did a lot of the research in the last, this last increment. It was around 350 different types of experiments. And for me, that's great because I like a lot of diversity of the scientific research. The one thing that I've noticed between 2002 and this expedition was the fact that I think the quantity and the caliber of the science has really dramatically improved. And I think we're using our capabilities on board the space station to a much higher degree, which I'm really happy to see. One of the really fun set of investigations that I did was tissue culture. And this particular one was an antibody drug conjugate that was being used to attack lung cancer cells. And it was fun to do because it's tissue culture, and everybody had slightly different apparatus. We were working inside the microgravity sciences glove box that helped us to maintain a sterile environment, or at least as much as we could in that environment. But I also enjoyed it because I got to use a microscope, and we were able to follow along and subjectively look at the fact that the antibody conjugates with the drug attached were killing the cancer cells. So it was pretty fun. I also looked at cardiac stem cells, and we did proliferation effects on different types of other types of stem cells as well. They all used similar types of technology. Again, we were working inside the glove box. We were trying to get a lot of work done inside the glove box because that was kind of our rate limiter for a lot of the investigations we were doing at the time. So it wasn't exactly a sterile environment because we were also doing rodent research in there at the same time. So we might do decontamination in between, but I wouldn't call it sterile. So I was very worried about whether or not I could maintain sterility when one day I'd be doing cell culture media changeouts and the next day I'd be doing rodent research. But we were able to get it all done. The cardiac stem cell studies, they've had some really interesting results. They're very excited about them. The folks also who are doing the proliferation of stem cells said they were seeing some really interesting differences in zero gravity, where proliferation was actually improved in some cases. So I think everybody's getting some really interesting results, and because I like tissue culture and I did tissue culture for a while, it was really exciting for me. We did some rodent studies. I mentioned that. We did several different ones, some looking at bone and drug testing to see if we can improve bone demineralization processes or regeneration processes. Here, the little black box in the top is a bone densitometer, so we were actually looking at effectiveness of other drugs on maintaining bone density during the mission. And other cell culture studies that we did in a non-MSG environment. And that worked to some degree. These are disposable little glove boxes, and they're on the right-hand side. And I think if we modified those disposable glove boxes a little bit, we could actually really improve our capability and expand the different types of research that we could do kind of laptop on the desktop, although this was done on the ceiling. But the types of research that we could do could potentially be expanded if we do some minor improvements on those glove boxes. We also did a lot of physical studies, physical sciences studies, including combustion studies. One of the things that you may not be aware of is that in zero gravity, when you burn something, when you use two fuels and make a flame, it'll burn out. But it's actually still burning, and we installed some specialized cameras that could even better detect this burning. They call it cool flames because it's not the same visual wavelength, and so they had to have special cameras to detect it. And even even that investigation showed some new results that they were not expecting because of the new cameras they had. So having the capability, this is a huge rack of hardware, gives you a generic capability, but having the specialized capability to include cameras that will focus on some particular aspect allowed these investigators to make some really new discoveries. One of my most favorite investigations, because I like working with plants and everything, so I got to grow some cabbage. It looks like lettuce, but they've called it cabbage. And we did that a couple of times, and both times we got to harvest it. Every other time we got to eat it, so that was great for the crew. We went to great efforts to make it special, so I would get some canned crab meat and make it into nice little salad that we would have on our lettuce leaf, because we only had 2 lettuce leaves each. So, but hopefully the investigators are getting some interesting research from the studies as well. But obviously, this is something that we need to have, this capability for our future long-duration missions beyond low Earth orbit in particular. Other experiments, we use this life sciences module rack, microscopic rack. In it, we grew plants in it, so they were looking at the initial germination phases of the plants and doing real-time studies using microscopy to look at those plants. We did protein crystallizations in here as well. So there's a lot of variety of hardware, these large capability hardwares that enable investigators to come in with pretty diverse experiments to look at different aspects of the effects of zero gravity, which I think is really important. But one of the other things that I think is important, and I think we really are using to a much fuller capability now, is that we have what's called the express racks, and they're much smaller racks, and the investigators bring them up, and every time a cargo vehicle arrived, we were rearranging basically the whole interior and setting it up for a set of investigations, which I was really impressed with, the fact that we were doing so much and so many different diverse investigations and using the express racks the way they were originally designed, which is to make it a plug-and-play type situation where, an investigator could bring up their study and we could work on it for a month and then send it home, and then the next cargo vehicle would bring another investigation, a completely different type. So I was really impressed with our agility and flexibility with that. This is the microscopy rack again. This is a little clip, a video clip that I wanted to show for one of the more complex human studies we were doing. You've probably heard about it, fluid shifts. And it involves a lot of different eye measurements and trying to understand the body and the changes that may be impacting our vision in space flight. But it's actually not a very non-trivial from an astronaut's perspective, because we have to haul a bunch of hardware, including ultrasounds and OCTs and different types of monitoring devices that measure pressure, and it's actually very complex, especially when we're doing it down in the Russian segment when we use the lower body negative pressure device. But it's actually much more doable than I gave it credit for, and I just wanted to show a short little video clip if you can punch play on that. And so this is me getting inside of the Chibis down in the Russian segment. We had brought everything down and set it up all in advance so that once we get in the Chibis, we're time limited. And we'd have to get all the hardware on, get all these measurements done inside of about an hour. So we were doing all different kinds of things, including ultrasound, and then OCT, which I thought might be very difficult as well, free floating. Okay, let's go to the next. And of course, we study a lot of biochemical aspects of the human body and of rodents, and we have to freeze them. We have these -80 freezers, which work pretty well. We had one that we had trouble with a couple of times, had to replace the electronics units, but luckily we were able to replace it without any risk to the samples. But we have, you can see where my knees are, there's half of this Melfi is showing you, so it takes up a whole rack, and we have 3 of those on board the space station. So it's quite a bit of storage volume, frozen storage volume, -80. We did a number of investigations. These are just with looking at how different computer programming will, for these spheres, to control and do different things like docking them together. We did also competitions with college students who had developed the software packages, and they would do competitions trying to take away prizes from each other using these 2 little spheres robots. You can see the blue and the orange, and then they have different adapters attached to them. Another experiment that we were trying, just because we're somewhat limited on our capability inside of glove boxes or maybe even the portable glove boxes, and some folks wanted to do a study in looking at surface tension and containment, and can we do more things just open benchtop. So we did some studies, and it's in a flat Petri plate with a large surface area on the bottom. We were able to keep, by shaking things laterally relative to the largest surface area, things you had to really, really shake hard to get anything to come out. But you didn't have to tap that hard on the bottom to make drops pop out. And so we were just doing some testing to try and better understand that and figure out what we can do in an open benchtop, because obviously tox levels of different things vary pretty dramatically, and we have to be thinking about that, what we can do open benchtop versus not. Another fun experiment that kind of was follow on from what Kate Rubens had started with the first sequencing using the biomolecule sequencer and a little PCR polymerase chain reaction device that we had on board. So I did some continuations of those studies, which worked out really well, but I was just really so impressed with the capability of just off-the-shelf hardware and how well it worked for these studies. And it was reproducible and reliable. They had the same samples on the ground and were getting the same results. And then kind of as a culminating effort, we took samples. I'd done surface microbial sampling and grown bacteria on orbit. And then I took a sample of that bacteria, amplified the DNA, and then sequenced it. And then we sent so I had the data on board and sent it down, and then we sent the samples down as well. And the ground checked it just to make sure that we had properly identified the bacteria on orbit. And I think, again, for those long-duration exploration missions, we obviously have to be thinking about capabilities that we're going to have to be able to do in-house, because on those missions, you're not obviously not going to be able to send the samples home to get the results. So you need to know and have those capabilities on board. So testing hardware that's off the shelf is not a bad thing. It does involve some risk because you never know exactly how fluids are going to work in space and in your specifics of your piece of hardware. But it's great when it works as well as the biomolecule sequencer and the PCR thing worked out. Over the course of the 3 expeditions we were up there, we loaded up the NanoRacks modules and did, I think, somewhere between 40 and 50 different satellite deploys during those expeditions, and I'm real impressed with the quantity of the research that we're launching off platforms aboard the ISS. And they are all different kinds of things. We also have done some 3D printing, which we did a lot of. So some of those were things that students had handed in for us to print. Some, like this, was a tool. They did some pretty elaborate things. And obviously, again, another technology that can be beneficial for us for those long duration missions in the future when we lose the one tool that we had and we need to fix something using that specific tool. So again, a very good capability for us to develop further in the future. And would you hit that once just to see if there's a video on this one?
Today we're going to talk about exercise in space.
This one is just showing you a little bit of the exercise hardware. I didn't know if everyone would be familiar with the
what's called a
resistive exercise
device. It's got a
device.
really important feature in order to protect the experiments that are going on board that might be sensitive to vibrations.
But I just wanted to show you the platform. And you can see the vibration isolation system I'm moving around. And again, that's obviously to protect the experiments that are sensitive to vibrations from our exercise. And since we're exercising 2.5 hours a day, it's obviously important to isolate our vibrations of exercise from the experiments. And this actually shows a very adaptable piece of hardware. It does take a little time in between, but it's very adaptable and flexible. And this is the first time I'd used this piece of hardware. The previous times we had an interim resistive exercise device, much simpler. Couldn't get the same weight or resistance. And so this, I just think, is an amazing, amazing piece of hardware. I put this slide in here. This investigation's already been published, but we were also doing other additional investigations looking at cardiovascular systems and oxidative stress, which I thought was an important study as well. And I think you've had some talks on some aspects of the cardiovascular system. At least I saw on your list of talks that you had talked about that a bit. Whoops, let's go back one. And also relative to the exercise, Jack was a subject for the SPRINT study, which is trying to use shorter, higher intensity exercise to see if we can reduce the exercise requirements required. And then I participated in some continuing studies looking at the biochemistry and bone density and nutritional aspects of life in space as well. Oops, go back one. We also have a ton of experiments that are going on externally, and all of these are ground controlled for the most part. But we've seen some really interesting studies with MAXI, other studies that look at winds on the Earth, temperatures, it's just amazing the number of studies. One of them that came up during my last increment was a study that was looking at neutron stars, and it was for basic research trying to understand neutron stars. But one of the kind of side studies that they had was neutron stars actually pulse at a very steady, very constant frequency. And they were developing and testing a software to see if you could potentially in the future on those long duration missions, when you are outside of where GPS will help you out, use neutron stars as your star positioning system as opposed to a global positioning system. And the initial results from that look very promising. They think they're going to be able to do that as well. And then I threw this slide in because I wanted you to, I'm sure all of you guys know, cargo, getting to and from the space station is obviously very important for our scientific research. For those future long duration missions to Mars, we're going to have to think about how we're going to get enough research there and maintain it while we're there, because obviously during my stay on this last time up there, we had I believe it was 6 different cargo vehicles. So it's, we were burning through a lot of the science that was coming up at that time. So we're going to need something to keep people entertained on their way to Mars. And while they're living there for a year, there'll be a lot of research obviously associated with Mars, but just thinking about research in terms of how are we going to do it from a very distant location and having all the appropriate supplies, capabilities that will enable it. On board space station, the psychological aspects are also very important. Being part of a team is really important. Again, for those Mars missions of the future, where we're much more isolated, that team camaraderie is extremely important. Team cohesion, very important. And I think the ability to look out the window and have your view mostly encompassed by seeing the Earth below it is going to be very different when we're on those missions where, we won't be able to see the Earth from below. I think it gives us a sense of connectedness that I think we'll be missing in those future missions. We also do some studies for global disaster warning, and so we take pictures from the station when there's something happening on Earth that has a global importance, whether it's flooding, hurricanes, whatever it might be. This wasn't Harvey, but because Harvey was much bigger than this. We got to see some interesting things with the solar eclipse. When it was passing over Earth, we could see the shadow on the Earth's surface. And punched the button here, and I think this is an aurora. Again, all of these things allow us to see the Earth and keep us connected, I think, to the Earth. And I think that's going to be an important aspect psychologically that we'll be missing on those future missions. So Earth's a beautiful place, and this is a time-lapse video of the Milky Way and if you imagine that this is all you're going to be seeing on those deep space travels, no solar ray, well, you might see your own solar rays, I guess, but go ahead and punch that and you'll be able to see that move. So this is it moving? Maybe the video is not working. Anyway, lots of stars. Oh, there it goes. There it goes. But I thought it was pretty neat that you could see, as we're moving 17,500 miles an hour around the Earth, you could see how the Milky Way was moving out one window. So, I think if we want to bring the lights up, we can take some questions, see what folks are interested in. I can't see anybody. We'll either have to bring the lights up or somebody will have to come forward to ask their questions. Or yell really loud. Okay, yeah. Crop in space. What would be my top 3 categories? One would probably be something like tomatoes, because it's a nice fresh thing that you can have, completely different than anything we get in all our rehydrated or MRE type foods. Cucumbers might also be a good one, because they also grow pretty easily as well. The lettuce was not bad. I like that. I'm thinking apple trees would be hard. So, if I were going to pick one fruit, probably orange. Something.
Plants would allow you to have that linkage when you couldn't see the Earth as a surrogate?
I think having plants has a psychological benefit, at least for those of us who enjoy plants. But I think even people who enjoy eating are going to like having plants and would enjoy the aspect of taking care of them. I don't know, it's not going to substitute for seeing the Earth, no, I don't think so. But I think it does maybe offer you a little bit more of home, in the sense that the diversity of the food thing is huge on orbit, in terms of, if you ask me what was the worst thing about flying in space, I'm going to tell you, as hard as those food lab guys work, and I cherish them tremendously, but the monotony of the food is probably one of the hardest things psychologically to deal with.
Thank you.
Mm-hmm.
Dr. Whitson, thank you for your contributions to space exploration and to science. In your last mission, you had a unique opportunity to witness the changeover of part of the habitat of the station. By that I mean the switching from the traditional fluorescent lighting system to the new tunable solid-state lighting system. I wonder if you would share with us your perspective and your experiences seeing both of these systems before the changeover is complete.
I definitely think the new solid-state lighting system is extremely better. I really liked, the different levels that you could set it at a lot, and in the mornings I'd put it on what the guys called Peggy bright, because they weren't as fond of it as I was. But I'm a morning person, so I liked having the really bright lights on in the morning. And then, in the evening when we'd do supper or whatever, we'd turn it down to the mood lighting for pre-sleep. So I found it very beneficial, and the lights are so much brighter that I do think it helps your mental attitude, and just to be able to have a lot of lights. On my previous expeditions, we had a lot of lights that wouldn't work, and so it was darker than normal. And with the vision changes that we have as well, having bright lights around is beneficial in order to try and see small things, when your eyes aren't working quite as well as they normally do.
Thank you.
Thank you very much for speaking with us today. I study team composition and teamwork and team performance, and you're well regarded for many things, but one of the things I hear you're really good at is bringing a team together and team cohesion. I'm wondering, what types of behaviors do you think can be done when you're in space as either the leader or just a crew member to bring the team together when they're falling apart?
Well, luckily I've never had a team that was totally falling apart, but we do quite a bit of training in the expeditionary crew skills, which the psych folks and I help them develop our training for that. And I think, obviously a very key aspect of those skills is communication. And we talk about how to debrief effectively and constructively so that, things don't build up and you don't develop tensions. But being a proactive follower, how to contribute as a follower and take some of the load off of the leader, making sure that the team is always, number one priority, and the success, makes us all that much more successful, I think.
Dr. Whitson, my question has to do with, as I watched you doing the exercise, and you talked about the importance of the exercise, and this was your third mission, and over the years, you're ageless, but as we age, and what have you found was the trick for you coming back? Because at each mission, you figure out, how do I get my sea legs back quickly so I can get back? What was a trick for you to now be able, after your third, to get back quickly?
I think my advice to folks is that initially you're going to feel a lot of the neurovestibular effects. You feel the weight of your body that you haven't felt for months. And, readjusting to that is not the most fun part. You feel sick, and if you just lie down, you feel better. But you still will feel sick when you get up. And so my advice to folks is get up, move around, force yourself to feel a little uncomfortable today because it's going to make you feel better tomorrow. So to push through some of that initial phase of just not feeling well. And obviously, having good drugs helps, to keep the vomiting level down. And some folks don't have that much problem. I would say in all the astronauts I've seen, I'm near the worst end of feeling bad when I first return, so I'm relying a lot on the drugs for the antiemetics. I rely on the IVs to increase the fluid volume, and I recommend that to everybody as well, because I've seen people who were actually pretty good at landing, and then 6 hours after they got off the plane in Prestwick, were then throwing up. And I think some of that has to do with that delayed effect of that lower plasma volume. So I really think taking the IV fluids is also really important upon landing, because we already know scientifically that you're going to have much lower plasma volume. It's going to take some time to redistribute in all the right places, but it's still, I think, a good first step as well.
Hi, thank you for your talk and for inspiring a generation of young scientists worldwide, including over in Europe. So my question today is actually, if you could have a wish list of technical/scientific capabilities, one thing you could have up on station, a technical or a scientific capability, and one thing for personal well-being that you could take up or have on station with you, then what would be your two choices?
Well, from a technical perspective, I think that's going to be an ever-changing thing, like the whole PCR and sequencing capability. I think that's a great step, and if we can keep taking steps like that to give us more and more real-time capability on orbit, I think that's important. for those future exploration missions in particular. For well-being, personal well-being, having been in space a few times, you know, lots of people sent me lots of things on my first flight, and, you know, after the first flight, I didn't allow anybody to send me anything that wasn't flat, like they can send me a card or a letter, or a picture. But other than that, I was picking the food that was going to get sent to me because I wanted a choice, and I didn't want anybody else picking my food for me. I wanted to pick the food I was getting sent. So,
Thank you.
okay, well, it's really great getting a chance to talk to you guys, and I really appreciate all the work that you're doing for our research and to get us to Mars sooner.